Hi, Stephen. Thanks so much for joining me today. I really appreciate it.
Yeah, absolutely. Thank you for inviting me.
To start, can you walk us through your journey into chemical engineering and what drew you to membrane science and separations?
Yeah, so this will probably be the longest winded answer that I give with all of this, but so if you go back to like my high school self and what I really wanted to do, I'm a huge sports fan. And I like as a freshman in high school, I remember telling people like I'm gonna go be a general manager of a sports team. Right. Like that was what I wanted to do. Build a sports team, make it be successful, right? Win all the championships. Growing up, at least the second half of my childhood in Kingsport, Tennessee, so a smallish town in the northeast tip of Tennessee. There, you don't have those connections to sort of build your way up that ladder to that so often. High school progressed. I really enjoyed chemistry and I was really good at math. And as I hear from a lot of the students that I teach, they're like, let's just put those things together and let's go study chemical engineering. And so that I was like, all right, sure, that's fine. Ended up applying to and getting in and then going to the University of Kentucky to get my BS in chemical engineering. What sort of got me into just the research in general is I did a co-op, which is basically going to work as your school for a few semesters. And in there, I got to work on a co-op with a research institute affiliated with the University of Kentucky. And so they happened to put me on they had this ceramic membrane project looking for coal fired power plant applications. And so that sort of got me into like, what is bench scale research and how to do presentations and read papers and all these sorts of things. And so I really liked that. Part of what made that interest more interesting to me is that right before I did my co op, I did a summer internship with a company that I didn't enjoy as much. And so I was like, oh, well, I really like this research. I want to keep doing this. And so I contacted one of the faculty at the University of Kentucky who happens to be like a world renowned professor especially in membrane science. And so that happens to be Dr. Deepakar Bhattacharya, or DB for those who know him. And if you've ever met him, you won't forget it. He's the life of every conversation. And he was a great mentor to be able to learn from. And his topic again was water treatment based membranes, which is what I was interested in, but using polymers as opposed to these ceramic things that I was utilizing in my co-op. And so I enjoyed that and was like, all right, I want to go to graduate school to do this more. And so I applied to a number of PhD programs and ended up choosing Clemson University to go work with Scott Hewson. And there I got to work on anti-fouling membranes using chemistry and surface patterns. And so I did that. And a sort of a funny story is going into that and most of my time there, my goal was to go work for some membrane company, whichever one wanted to hire me at the time would have been what I wanted to do. Probably was go work for Dow, which is became Dow DuPont, and now DuPont, which is where the film tech, the membrane arm of that is. And then, sort of the last year or so, I got pushed by both my wife and another friend of like, you'd be a really good professor, like doing these things. And I was like, I would like to pick my research problems that I work on and continue to get to mentor these different students and things like that. And so I quickly pivoted to like, all right, I want to go be a professor now. And I went through the interview cycle and happened to get hired here at the University of Alabama right away upon graduating. And I've been here ever since. So now I've been here 8.5 years, now associate professor in the Department of Chemical and Biological Engineering and working on all sorts of water treatment problems, some membrane solutions, but some also not. Some we're using solvents too, and some polymer absorbers. So all sorts of fun things.
Awesome. I appreciate you telling us about your background.
Yeah.
I actually had similar aspirations in high school, but I actually carried it through to studying sport
Oh,
management
awesome.
at Western New England University.
Yeah. Oh,
So
great.
the two things I had thought about doing when I was in high school was I was interested in communications and radio,
Mm-hmm.
and part of that I'm kind of getting to do some of with this
Sure.
podcast,
Sure.
which is pretty cool.
Yeah, that's great.
Your work focuses on membrane functionalization for water treatment. How do you view the role of membrane separation in broader analytical chemistry workflows like chromatography?
Yeah. So I think it depends, right? When we talk about membrane treatment, there is a wide variety of options out there. And what I always tell my students, and then, in classes, I often teach a separations class, or I've sometimes taught a more water and wastewater treatment focused class. I always tell them, like, it depends on what you have coming in and what you want going out. And so, depending on those applications, will depend on what type of membrane you might want to pick, if a membrane is even the right option for what you're trying to do. Right. And so, like, a membrane can certainly be very useful in concentrating things if you're trying to dewater systems or some other solvent. As long as it's compatible with the solvent, it's certainly easy to try and bump up the concentration. Those are pretty standard things for us to do. That's essentially what we're doing when we're purifying water anyways. We are dewatering systems. It's just we're producing the clean water that you want to have and concentrating the stuff that you don't want in a separate stream. Pre-treatment's a big one, right? If there's a particular contaminant or something that you don't want in there that we can do with a simple membrane filtration, then we'll just do that. That's not overly complicated. If you've done syringe filters, for those out there, right, you use a syringe filter, that's a membrane more often than not. And so you're doing that pre-treatment step automatically. And even with dust and debris, sometimes that becomes a problem, right? You can use membranes to filter that out too.
You've been around membranes for more than a decade at this point.
It's crazy to think.
What are some of the most transformative advances that you've seen in membrane materials during your time studying this area?
Yeah, I mean, I think the past few years, you started to see, and it's now much more commercial, is like these, what they would call lower pressure reverse osmosis membranes. There are sort of two classes of very selective membranes called nanofiltration and reverse osmosis. Typically, they're similar chemistries, but slightly different. And one, reverse osmosis, or RO, you've probably heard of for like seawater desalination, right? That's where it's most commonly used. But nanofiltration is also helpful, especially for like hard water applications. It's really good at removing divalent ions, but still allowing those monovalent ions to pass through. And so part of the difference is nanofiltration would allow more water flow through the membrane at lower pressures, where reverse osmosis typically requires the higher pressures of the membrane systems. And so there's been a lot of work done by the membrane industry to reduce the pressure burden on reverse osmosis membranes. There's been pressure recovery devices and all sorts of things, but that's not really the membrane core technology, developing those lower pressure reverse osmosis membranes. So you can either use lower pressure to produce the same amount of flow or reduce the amount of membrane area, so you reduce your plant footprint, has been a big deal in those spaces. And then, in the research community, there's tons of awesome research being done out there all over the world, making membrane adsorbers so that we can get a little bit away from, like, we'll call it membrane chromatography in some sense. So we're trying to do these chromatography separations that you typically do with a column, but now we can do it with a membrane, and oftentimes they become faster separations and we can do higher throughputs. That's taken off more in the protein and biologic purification industry as opposed to some of the more water treatment industry. I suspect as the demand comes for more selective separations, especially for certain elements and minerals and molecules and things like that, I hope to see that more commercialized as we try to move forward.
I always associated reverse osmosis with Dasani water
Sure.
because that was the first time I
Yeah.
had ever
Yeah.
heard that phrase. And
Yeah.
now I have a unit installed under the sink
Sure.
by my landlord that
Yeah. I mean, seawater desalination just in general is now dominated by RO, whereas it used to be thermal evaporation, which is much more energy costly than reverse osmosis.
in your previous interview on the Noon Show,
Mhm.
you discussed water quality and membrane filtration basics.
Yeah.
And how do you approach selectivity in membrane design for specific target compounds?
Yeah, it all sort of depends on what you're trying to achieve with your desired separation. Are you wanting to capture that or be able to concentrate a specific material, or are you trying to remove certain classes of material? Some membranes separate almost exclusively on size, so those would be things like microfiltration and ultrafiltration. There are some charge dynamics within that, so it's certainly not negligible, but most of it's done based on size. And so if you have certain size differences, then you can choose an appropriate membrane for that. And there are tons of different membrane options that have different pore sizes, different pore size distributions, different chemistries to be able to choose the right membrane for the right application. When it comes to things like salts and PFAS, it depends. Can you tolerate some sodium chloride? If so, you should use a nanofiltration membrane. If not, right, then you should use a reverse osmosis membrane. PFAS, most of the PFAS can be rejected by a nanofiltration membrane. Maybe some of the smaller molecules might be problematic. Some of those shorter chains are a little more challenging with nanofiltration, but for the most part, nanofiltration should be doable to remove most of that. So it really just depends on where you want certain things to go. In a membrane system, you have your feed coming in, your membrane processes some amount of water, you get a product stream or what we would call a permeate stream. Typically, when we think about producing clean water, that's our clean water stream. But then you also have what could be called the retentate or the concentrate, essentially the reject stream. And that is what didn't pass through the membrane. So that'll be some amount of water and then everything that didn't go through, which will be much more concentrated than it was in the initial feed stream. And so you have to think about like, well, what is in that stream? What do we do with that stream? Because it's now generally a waste stream, or is there something else that you can get out of it that we need to further do more processing on, or can we discard that back to the environment with some treatment, or just kind of depends on what the system is and what you have in there.
We certainly talk about PFAS a good amount on this podcast
Yeah.
because it's certainly the number one issue at the moment in terms of environmental contaminants in general. Slight plug for my company, Organimation. Our evaporators are used in a couple of the drinking water quality methods.
Mm-hmm.
What we're doing is the evaporation of dryness after the solid phase extraction
Okay.
ahead of LCMS.
Yeah.
What role does fundamental separation science like chromatography play in verifying membrane performance or validating contaminant?
Yeah, so I mean, it depends. Chromatography. It'll depend on what the analytes are that you're trying to deal with. One of the things that we end up using we use ion chromatography for specific ions of interest. Sometimes ICP works, but sometimes it doesn't. Ion chromatography is certainly one that we've been looking at. There are others. We've been using GCMS and electrospray ionization for certain molecules because it's a little bit easier to detect that way. Like, for example, the most recent paper out of my group was attaching an enzyme to a membrane to make it what we would call a biocatalytic membrane. We were using that to degrade phthalates, which are additives used in plastics. We were using electrospray ionization GCMS to be able to detect both the initial phthalate as well as its degradation products. And so that was really helpful for us to be able to do that. It kind of just depends on what you're doing and whether it can be useful or not. I know purification columns are useful for biologics and protein separations and things like that. It really depends on what you're after, right? Certain, your simple basic, again, like desalination salts, chromatography in general isn't maybe the most useful, but there are a lot of newer emerging separations where it's vital. Again, things like ion separations, it becomes really important.
Yeah, this recently just came up in a question I was asked. And in terms of the name of the podcast, we called it Concentrating on Chromatography, partially because of that alliteration, but we focus on mass spec and separation science in general.
Yeah, I mean, it's all, chromatography. And again, you can call some things membrane chromatography too. So it's a broad term that fits a lot of different things.
Some academic separation technologies struggle to scale well. What challenges have you encountered in translating membrane research to industrial or maybe municipal application?
Yeah, that's a very loaded question. Probably I would say the number one, well, there's two main problem or challenges with this. One is always time. The time it takes to do certain reactions or whatever you're trying to do often is not practical for an industrial scale. And oftentimes we don't always, as academics, optimize that to get it as low of time as possible, because as long as we find something that works, we kind of tend to just stick with that and run with it. Whereas industries like, can we do this in a minute? Can we do this in 30 seconds? Can we do this in 15 seconds? Sometimes that's not super practical for us to do on a bench scale. And the other one's obviously cost, right? Certain chemical costs. And it's hard for us to project some of that because we're often not privy to bulk chemical costs, or it's not something we necessarily look at. So, why something might be expensive for us perhaps actually isn't so expensive when you're buying a drum of it or 20 kilograms of it. That's more than we would ever use on these types of things. So, those are the two biggest challenges, I would say, with that. But part of that is, the funding really isn't there for us as academics to push the research in that direction well. There are certain places that you can try to get some of that, but it's really hard to go from, like, yeah, I have this technology, it works on a bench scale, maybe a small continuous process that you've developed on the bench, to move to a pilot scale. It requires a lot of time investment to get all of that to work out. And that becomes really challenging. And again, that oftentimes ends up being outside of the mission of what a lot of us try to do, which is educating students on fundamental research topics. And while scale up certainly could be a part of that, it's not always the quote sexiest idea that students even want to work on. They want to do the fundamental science part and make these new discoveries, not like, well, how do we scale this up to the next step? And so that becomes challenging, and oftentimes that's also expensive. And so, getting the appropriate amount of funding to develop it from there farther is challenging. So we'll submit things like IP disclosures and things like that. And, the holy grail is hoping someone wants to take it and go with it. Maybe we consult on how to process that, but, oftentimes that really doesn't happen. I think, our role, to me, is to lay the groundwork for most things, and then, someone hopefully gets excited about it and wants to pick it up and move it forward from there. And sometimes that's conducive to creating your own small company to push it forward. It kind of depends on the technology as to whether you can do that or not. But I also think it's also important to have this mindset of like, well, what is scalable, thinking about it from the beginning, right? We always have to think about the fundamental science aspects, but we have a funded project from the National Science Foundation right now that is looking at reverse osmosis membranes, and we're not trying to improve seawater desalination. We're looking at small molecule rejection. So as an example, like urea, these uncharged molecules, or boron is probably something that you've heard of with that. And so we're trying to see if we can use some additives within the membrane fabrication process to improve that. And so our rationale on that point was, well, we're not changing how they make the RO membranes. This would just be simply putting an additive into their process. And so to me, like, of the things that I've done so far, that's probably the most scalable, because it's just a matter of, can you buy the surfactant, and the company consider that of reasonable cost to be able to do that, right? Whereas, like, we were also doing that with RO membranes, but trying to modify already commercial products. And, that would take us sometimes 1 day, 2 days just to do the full modification. And I'm like, company's never going to pick that up. So there's challenges with some of that. So I have thought about that on some degree, but, it just kind of depends on what you're trying to do.
That always interested me as a representative from a small business manufacturing company where I attend a fair amount of academic conferences, and you'll see a poster presentation where the title starts with a low cost product solution for X. And I always wondered if there would be a way to help partner with that commercialization effort with the researcher. It definitely seems like there's an opportunity there, but I suppose so much depends on the specific project, but more how ready it is to go to market. Where are they at a spot where they're months away from sending out designs for production,
Sure.
or where in the development process they are? But it always seems like an open opportunity to me.
Yeah, absolutely. And scale is a huge deal. Like, well, what scale do you need to produce? What are you currently producing? Oftentimes, in terms of membranes, we often produce maybe a sheet of paper size. And when you're talking about making a membrane system, you're talking about 40 feet wide by more than 15 feet long. And so, how those things scale is very different. And so, how you make those is very different. And so, there's got to be research in these more continuous processes that do that, but it also becomes a lot harder on us. We have finite lab space, and we've already got some systems in place that they're also expensive. So you've got to get funding to be able to pay for all that. So we try on certain aspects, and other aspects, that like, yeah, this part's not scalable at the moment, but maybe it will be, or if this works really well, now we'll start thinking about that. Maybe we'll partner with somebody, again, get some industry involvement and guidance on what we need to do to get that to work.
Certainly, labs becoming more green or sustainability in general are kind of evergreen hot
Sure.
topics. In the next 5 to 10 years, how do you see that impacting the field of separations or membrane science in general?
Yeah, I mean, there's certainly pushes for more sustainable membrane manufacturing, right? Membranes as a process are pretty green, relatively low energy requirements, and things like that. So, in terms of as a process itself, they're not too bad. And lifetimes of, a certain membrane element will probably last. Typically, it's advertised about 5 years or so. So it's not too bad of a lifetime with that. You never want a perfect membrane, because if you ever made a perfect membrane that you never have to replace, you're never going to make money. So, you know, 5 years seems to be a nice sweet spot that people have found. But where it can be better is on the manufacturing of the membranes themselves. They often use hazardous petroleum derived solvents. And so some of our work has been in that and trying to use greener solvents, right? More bio based solvents. And there's lots of good people out there doing all sorts of good work on that area. We're doing a little piece of that. The other thing we're actively involved in is trying to look at using waste plastics within membrane manufacturing. These plastics are already out there, often contaminating the environment at this point. So, can we give them a second life so that, if nothing else, we've just increased the lifetime till they end up back in the environment? Ideally, right, perhaps looking at alternative polymers that are more biodegradable, is probably less petroleum derived, is probably better on the sustainable side of things. But again, a lot of that comes down to cost and do they perform the same way? I think one of the big discussions, it's not so much happening here, but that's happening more in Europe, is, do you consider polyvinylidene difluoride, PVDF, as a PFAS? And PVDF is, if not the largest, one of the largest polymers used in membrane separations. If you consider that a PFAS, that becomes a huge change in regulations, and that would be disastrous to a number of companies. A lot of people, users of the membranes, are now going to have to change to different membrane systems that may not perform quite as well. And so I hope that, for me personally, I don't think that's true. I don't think you should consider PVDF as a PFAS, although I understand the argument for it. But I hope that ends up not being regulated as such.
My understanding is most of the US EPA standardized methods for drinking water for PFAS were developed about 5-ish years
Yeah.
ago. But it really sounds like right now, last year, this year is when Europe's having their big regulation push. That's just my understanding of it.
Yeah, and a lot of what we tend to see is we lag a little bit behind Europe. So once Europe, whatever regulation, we tend to follow suit a little bit later. So, like, as an example, one of the solvents that's used in membrane manufacturing is NMP, N-methylpyrrolidone, and the EU has since banned it, and it's not used really as much anymore in commercial membrane manufacturing. But a part of it too, right, is these industries, they're not going to, and I don't fault them for this at all. They're not going to change how they make their product until they have to by regulations, right? So, we talk about, like, oh, we've got this new green solvent. Why don't you guys use it? And they're like, well, we're not going to until we have to, which is totally understandable, right? They have a product that works. It works the way they want it to. And until they get told, you need to start thinking of, like, this is imminent, you need to start thinking about how you're going to change it, right? They're probably not going to, unless you had them, like, you'd have to present a lot of evidence, cost, performance, reproducibility, time, to be able to convince them otherwise.
I mean, it's kind of unrelated, but the one that was always a head scratcher for me as an outsider was the somewhat ban on methylene chloride. It's banned, but also it's used in all these methods, which, if you're doing those methods, guess what? you're going to have to use this.
Yeah, I mean, the simplest pivot to that is to use chloroform, but it doesn't work always the same. I usually call it dichloromethane, but same thing. There are certain types that's just what you should use because it works better. And now you basically, at least here, it's basically like no chance, don't use it. If you need to use it, you have to get all this approval, you have to wear all this safety gear. At the end of the day, it just becomes too much of a headache.
It was interesting to me earlier when you mentioned Dow. Most of my experience with membranes has been Corning.
Mm-hmm.
We have an accelerated one step extraction unit, which basically combines liquid liquid extraction and then the concentration
Mm-hmm.
step. So, old school chemistry,
Yeah,
but we've been reselling those membranes for that instrument for
cool.
decades
Yeah,
at this point.
yeah. Yeah, we've been doing some fun liquid extraction systems for what we would call hypersaline brine desalination. And so this is not like, don't think seawater, think like much saltier than seawater, perhaps 3 times saltier than seawater. And so in this case, it's sort of the opposite of a typical liquid extraction system. Typically, you try to pull the salt out of water. And in this case, we're trying to pull the water out of the salt water. This has been a really fun project to learn about. And we're still just exploring some of the beginnings of how these things work.
And then, way on the other end, we have our nitrogen generators, which
Mhm.
are real simple membranes where it's compressed air in and nitrogen out. And but obviously, it's a product that solves a lot of headaches. Where I'm not sure if you rent the gas cylinders for your lab or what your nitrogen
Yeah,
setup
yeah.
is, but
Yeah,
membranes really save the day
yeah,
there.
absolutely. And even if you need like oxygen enrichment, especially when you think about the medical community needing it a little bit enhanced oxygen, using those air purification membranes to be able to separate some nitrogen and oxygen and concentrate it a little bit is those are a neat invention that's happened out of the membrane community.
You touched on it earlier, but how do you mentor students with needing to balance that deep fundamental research, but also having the application of the applied problem solving aspect?
Yeah, I think we have to have an application in mind, and as a sort of membrane scientist, as I would typically call myself, that's not overly challenging, right? There's a separation that we're trying to do of some sort, and so we're trying to make things a little bit more sustainable. What is our overall goal? And then you look at, all right, well, what are the different sub pieces as a part of that we want to achieve? And so then we break that down to different pieces that we're trying to look at, different sub hypotheses, different research questions we're trying to ask. You're still keeping that goal in mind, but then trying to answer those very specific fundamental questions. Then what is that fundamental science do we need to do? What are the experiments? What are the characterizations? Do we need to get some modeling involved to try and understand how to do some of this? So it really, we always keeping the application on the forefront of our minds, but then absolutely, we've got to dig into certain fundamentals, and sometimes that doesn't really matter so much on the application unless we're talking about performance results, but more of like, did we make what we were trying to make? We were trying to modify the membrane with some chemistry. Did that happen? Yes or no? How did it modify it? How much did it modify it? Do we still have an okay membrane? Is it still permeating water at a good rate or not? Yeah, it's always a balance. And sometimes you got to slow things down like, hey, like, no, I'm a big proponent of like our controls have to be good. And if our controls aren't good, people aren't going to believe the rest of the data. And so even if it tells a cool story, right? If, as an example, if we produce an RO membrane and we have 85% sodium chloride rejection, well, who cares? Anybody can improve on that. So we've got to make sure that our initial membrane before we do anything to it is good. And then once it is, right, then we can start doing all sorts of things from there. That's when we get to do the fun part. And it may take 3, 4, 5, 6 months to get the control. And that's really boring for a student because they want to do the fun science. But I'm like, until you do this, right, then we won't know that it's better or good. And so we've got to make sure we understand that well.
Absolutely. That makes sense. Was there anything else you wanted to talk about today, Stephen?
We talk about the research funding. Certainly, this past year has been really chaotic on our front. There's been less calls, very prolonged timeframes of getting decisions on when, if proposals are funded or not. Luckily, it seems to be that things are opening up at least a little bit. Hopefully, we'll get some more calls. Of course, now things are getting pushed more towards administration priorities, which is okay. And we just have to learn as academics. That environment can change every 4 years. You never know. The ability for us to be able to pivot and do things like that, I think, is important. And some of that too, right, is, of course, all of us have said that. Like, it'd be great to be able to get more industrial funding to be able to do things, right? And, industry only has so much money and they're not going to spend it all on academic research that, as we mentioned with translation, oftentimes doesn't always lead to something that's useful for them. That's an interesting balance that we have to play. But at the end of the day, our job as academics, to me at least, is educating students, and that's undergraduate and graduate level, right? We want to make sure that we create graduate students that are good researchers that will then benefit, your companies. Even if things like, hey, we have this problem, we need to solve it. Here's some information, right? You don't have to give super specifics on whatever the separation is. Some information to be like, hey, here's a problem, go work on this, guys. And we can seek out funding in this area and then say, hey, like, we'll connect with this collaborators and maybe get a letter of like, hey, yeah, we support this idea, even though we're not putting any money forward. And those types of things can be really helpful.
Yeah, I figured I would let you come back to that topic if you wanted to discuss it more because I know it is so touchy. It's hard to talk about.
Yeah, I mean, the biggest thing for me is really making sure that I can support my students in my lab to make sure that they're able to graduate, right? That, whether I get some of my salary covered or not, I don't really care. That doesn't matter as much as it is to make sure that I have money to pay my students. And at the end of the day, that's the important part. And if we, nationally or so, it was about a 10% funding rate prior to 2025. That number seems to be cut in roughly half. And so that's going to make it harder to be able to fund the number of students that you have, right? It's like I have 5 PhD students in my lab right now. May not be able to do that in 2-3 years. May have to get cut down to 3 and kind of just go from there. And so that also, at the end, in the long term, that'll hurt lab continuity. One of the nice things about having 4, 5, 6 PhD students is ideally you have one or maybe one a year or so. And so as they become more senior, they train the more junior graduate students, and you get this rolling cycle of continuous knowledge retainment within the lab. And so if that's more spaced out, that may not always be as possible to be able to do some of that stuff. And so there may be longer term ramifications that aren't happening now, but might be happening in some of our labs in 2-3 years.
Yeah, previously I spoke with Guy Caldwell, who you may know from your university, just some of the lengths that he and his wife have gone through to make sure their lab and Department of Biological
Sure.
Sciences, the lengths they've gone through to make sure that they can in their head count. It's just amazing what he's done, what they've done, what you've done to put the emphasis on making sure that you can keep paying these students to do their great work they're doing.
Yeah, and it's work experience for the students. Even if you're still in a degree program, right? To me, if you get takes 5 years to do your PhD, that's 5 years of work experience you've got learning how to do research for a company, and so that it's very valuable. And the cost isn't a ton, right? PhD students in chemical engineering, again, it's going to depend on the university and the department as to how much they make, and including tuition and health insurance and fees and things like this. But it's not a ton, so it's just a matter of figuring out where does the money come from to get that taken care of. Because they're awesome and they're bright and they're fun to work with and they're energetic and they got cool ideas, right? And we want, we you don't want to kill that because we can't fund them, right? That's again, part of my number one job with doing these things is making sure that we can pay the students to continue to work.
Well, Stephen, I really appreciate everything you've been willing to share today. Thank you so much for your time, and I can't wait to share this episode with the audience.
Yeah, awesome. Thank you. I really appreciate the invitation. It was great of Lisa to connect us. It's been my pleasure. So thank you so much.